Hello, my name is Jessica Escobar, and today I will be sharing my honors research presentation, which is titled Investigating the Role of Zinc Finger Protein 30C in the Transcriptional Repression of Drosophila engrailed gene. And so before we get into the specifics of the research, I want to go over DNA and how it's packaged and repressed, etc. So within the nucleus, DNA exists as chromatin, where the DNA molecule is tightly wrapped around a core of histone proteins and usually remains in a silent form, which is called heterochromatin. And so for a particular gene to be expressed, the chromatin opens into an active conformation, which is called euchromatin, and this allows the transcription machinery to reach the target gene. And so today we're going to talk about epigenetic control of gene expression. And so open chromatin is changed to a closed chromatin state by DNA methylation and histone modification, and that's indicated by the red circles and blue triangles shown in Figure 2. And the closed or repressed state prevents transcription, which is called epigenetic regulation. And the repressive histone marks are placed by Polycomb group proteins, which is a specific group of proteins that we're focusing on in this research. And so different sets of genes are turned on in different types of cells. The ability to activate or turn on and the reverse, silence and turn off transcription of specific genes allows for differential gene expression and thereby enables cells to have different fates. Holistically, the loss of this regulation and control can lead to cancer. So here are Polycomb group proteins. These proteins modify chromatin to repress gene expression, and they're required for differentiation and the maintenance of cell fate. Polycomb group proteins are organized into multi-protein complexes called PRC1 and PRC2, and these PRC1 and PRC2 have different biochemical activities towards chromatin. PRC1 leads to ubiquitination, and PRC2 is responsible for putting on the repressive H3K27Me3 mark. In this study, we focus on H3K27 methyltransferase, which is a histone-modifying protein and catalyzes the transfer of methyl groups to lysine and arginine residues of histone proteins. Essentially, this is a post-translational modification and thereby turns the DNA off by causing the histone to encompass the DNA with its tail. And so in order for Polycomb group proteins to and for the specific region that we focused on, we're talking about engrailed, and engrailed expression is regulated by PCG genes. And so the protein engrailed sets up the anterior and posterior boundary of each segment of the fly embryo, and it's expressed in the posterior part of the segments. And so a mutation in the polyhomeotic gene causes engrailed to be expressed in regions where it should not be. And engrailed is expressed in the posterior part of every pair segment, so the expression is turned off in the anterior part. However, mutations cause expression all over the pair segment, which is shown in Figure 6. Figure 7 shows the upstream region of engrailed, which contains two PREs. Here we focus on PRE2, which is a 181 base pair long fragment that contains two sites labeled A and B with unknown DNA binding proteins. When the binding site displayed on the slide was mutated, the PRE was no longer functional. And so this magnified region was used as a bait to identify proteins that bind to site A, which we talked about in Figure 5. We're trying to find DNA binding proteins that are present at the PRE. And so candidate proteins were found by DNA affinity pull down coupled with mass spec. And so essentially what this is using biotin on the sequence of 18 DNA pairs, which were shown in the previous slide. Then nuclear extract was added because this is where transcription factors are, which is in the nucleus. And this was done for the oligo and the mutated region. Then it was incubated for 2 hours at 4 degrees, and beads were added which were coated with streptavidin, which specifically recognizes biotin. Wherever biotin is, streptavidin will bind because the beads are magnetic. and then DNase was used to degrade the DNA and release proteins to be identified by the mass spec. And this leads me into my statement of purpose. The purpose of studying transcriptional repression and its regulation is to build an understanding of how these processes relate to developmental pathways in cancer. We are using the common fruit fly, Drosophila, as our model system to understand the mechanism of silencing of a developmentally essential gene engrailed by a class of transcriptional repressors, Polycomb group proteins. We are investigating the role of zinc finger protein ZF30C in the recruitment of the Polycomb group proteins and subsequent repression of engrailed gene because loss of potential ZF30C binding site led to a loss of repression at the engrailed locus. And this leads me to my thesis, which is that zinc finger protein 30C is a DNA binding protein that specifically binds to site A and PRE2, which was deemed critical for transcriptional repression at the engrailed locus. So I want to cover a little bit about zinc finger protein 30C, which is our protein of interest. ZF30C was implicated in controlling receptor gene transcription in sensory neurons and wing patterns. It has 13 zinc fingers, and these are essential for DNA binding. It has a high-level expression in embryos, and which is a characteristic of transcriptional repressors involved in engrailed silencing. And lastly, it's a C2H2 zinc finger protein, which obtains its name from the binding of zinc ions to cysteine and histidine residues. So our first approach in revealing the presence of ZF30C at PREs was using polytene squash. Polytene chromosomes are giant chromosomes that begin as normal chromosomes, but through repeated rounds of DNA replication without any cell division, they become large banded chromosomes. These chromosomes provide multiple copies of a protein binding site, and therefore binding of a protein can be easily visualized. The methods of this were, I collected the salivary glands from third instar Drosophila larvae, which is indicated in the red oval, and it was expressing GFP tag ZF30C. I then chemically fixed them on a slide and used a pencil eraser to squash the chromosomes in the glands. Once I verified via microscopy that the chromosomes were squashed, I stained the chromosomes on the slide using anti-GFP antibody. Additionally, the chromosomes were co-stained with antibodies against the well-known PRE DNA binding protein, Pho. The purpose of this technique is to see via fluorescent microscopy if ZF30C is present at PREs in conjunction with Pho, a DNA binding protein present at the PREs in Drosophila genome-wide. So we can see in figure 10 that ZF30C co-localizes with Pho at genome-wide PREs. The first column shows the chromatin in DAPI, and then next to it is ZF30C in green, Pho in red, and then the merged region. And so overlapping regions are represented by yellow bands indicating a co-localization of ZF30C and Pho at PREs. I want to emphasize specific binding that's occurring, and that's validated by the fact that some bands are only green and some bands are only red, indicating that ZF30C is binding to specific PREs and nonspecific signaling is unlikely. We tried to prevent nonspecific binding and signaling in our procedures by using blocking buffer. So now that we know that ZF30C shows partial colocalization with Pho on several PREs in the Drosophila genome, we want to investigate if it is specifically present at the engrailed PRE. And so in order to do this, we used chromatin immunoprecipitation to investigate the presence of ZF30C at engrailed gene. And chromatin immunoprecipitation is a technique used to investigate the interactions between proteins and DNA. If you look at figure 11, this is the mechanism of CHIP, otherwise known as chromatin immunoprecipitation. And so we're trying to see if ZF30C is localizing to this DNA. So in general, first is cross-linking a DNA binding protein to DNA. Then the chromatin is isolated by sonication, which shears the DNA that is bound with the protein into small fragments. Then an antibody is used to bind to the DNA binding protein to isolate the complex by precipitation. The DNA is then purified and proteins are digested. Lastly, we used PCR to amplify specific DNA sequences to see if they were precipitated with the antibody. And so specifically for this research, we used wing discs and leg discs from the ZF30C GFP third instar larvae, which were dissected and fixed. Then we used sonication, and the tissue was homogenized and the DNA was fragmented and then incubated with anti-GFP antibodies. Then the DNA was immunoprecipitated, purified, and analyzed by PCR to check for enrichment. PCR amplifies DNA and uses primers to specify regions of DNA. In this experiment, we used engrailed primers for specificity and viewed the size of the engrailed PRE DNA using gel electrophoresis and base pair measurements. We expect the DNA to be between 100 to 200 kilobase pairs, as PRE2 at engrailed is 181 base pairs long. So here we have the results of the chip. In lane 1, you can see that the DNA was amplified using primers for engrailed PRE, and then the control for it was next to it. In lane 2, we don't expect for PCGs to be binding here because it's the control. Then in lane 3, this represents the input DNA, which was collected before enrichment. Lane 1 and 3 show the expected product at a size of 150 base pairs, which is really close to what the engrailed PRE2 was. Then lanes 4 and 5 represent reactions with anti-IgG antibody, which is a control for nonspecific binding. Since IgG should not bind to engrailed PRE, we don't expect a product in these reactions. And so this gel reveals that ZF30C that the engrailed DNA was somewhere around 150 base pairs, and a lighter band for the ZF30C input represents that ZF30C was enriched. And so in conclusion, we can see from the polytene squash and chip that there's an identification of a novel DNA-binding protein, ZF30C, which binds to the engrailed PRE and regulates its expression. And so for future directions, we want to finish up chip by following up with qPCR from the endpoint PCR. qPCR tells the difference in copy numbers of DNA, and the enrichment causes more copies, which is shown in the number of cycles. So we expect more enrichment and more cycles and less cycles in our ZF30C versus our input. And then we'll follow up with ChIP-seq, which is specifically looking for the engrailed PRE, and we use ligation to add adapters. And then non-specifically add onto the 5' end DNA fragment, and then it goes on to sequencing so that we know everywhere where ZF30C is binding. And so lastly, I want to acknowledge, I went to make some acknowledgments. We acknowledge funding and the support from SC Embry, and then we also want to thank Billy Ardelt for allowing us to have the cold room and for setting that up. And then I also want to thank the PC Biology Department, first Dr. Ray, for helping me and being my advisor. I wouldn't have been able to do any of this without her, and my research would be incomplete without her. I want to thank Dr. Scholl for allowing us to use RIPA, which was used in our chip. And lastly, Dr. Gordon for allowing us to use his facilities and allowing me to spend some class time on this research. Thank you, and if you have any questions, please let me know. To sustain transcriptional repression, they have to be recruited somehow. And so Polycomb group proteins get recruited by DNA fragments called Polycomb repressive response elements, otherwise known as PREs. PREs contain binding sites for DNA-binding proteins that help to recruit the PRCs to the target genes. And here in this figure 5, you can see Pho is present. Pho is a DNA-binding protein with a DNA-binding domain and is found at PREs. And so Pho is necessary for PRC recruitment, but is assisted by other DNA-binding proteins. And that's where SPPS comes in this figure. SPPS represents a DNA-binding factor present at PREs. And the X next to it denotes the unidentified DNA-binding proteins, which we'll be investigating in this research.